discussed about the medical importance of the sugars. They are the source of energy, they serve as a reserve food material, and they are the structural components of many animal and plant structures, and they are the components in the cell membrane, and also the nucleic acids and human blood group substances are formed because of the minute change in the sugar residues present on the surface of the red blood cell membrane. And certain sugar derivatives are the antibiotics and also the drugs, and mainly the voavine. It is a modified cholesterol with the sugar molecule, and it is a sodium potassium ATPase inhibitor. It is also used as a drug for then we discussed about the classification of carbohydrates and the monosaccharides, oligosaccharides, and polysaccharides. Then we discussed about the classification of monosaccharides further based on the number of carbon atoms based on the functional group. Then we discussed about the D sugars and the L sugars, and most of the sugars naturally present, naturally abundant sugars are the D sugars, and the configuration of the D sugars based on the glyceraldehyde configuration. And in the glyceraldehyde, if the hydroxyl group on the chiral center is present on the right side, then it is called D-glyceraldehyde. If it is present on the left side, this is called L-glyceraldehyde. The configuration of all the sugars is based on the D-glyceraldehyde configuration. You can also see here, if the sugar has D-glyceraldehyde configuration on the penultimate carbon or the last but one carbon or the bottom stereocenter, it is known as the D sugar. If it has the hydroxyl group on the penultimate carbon on the left side, then it is known that they are known as the L sugars. So there are many tetroses, pentoses, and hexoses naturally abundant, and we have already said how to remember those structures. And then we also discussed about the various ketoses, and the simplest ketose is the dihydroxyacetone. It doesn't have the chiral center. When we incorporate one more carbon chiral center next to the keto functional group, then it initially it gives the D-erythrulose, and then incorporation of one more carbon next to the keto functional group and positioning hydroxyl group on once on the right side, once on the left side gives the D-ribulose and D-xylulose. From D-ribulose we get the psychose and the fructose, whereas from xylulose we get sorbose and tegatose after when we introduce one more chiral carbon. Then we discussed about the properties of monosaccharide, asymmetry, then the isomerism and the optical isomerism and the stereoisomerism. Then we also discussed about the what is the dextrorotatory and the levorotatory, and it is designated with the either small d or the plus sign. Levorotatory is designated with the small l or the minus sign. And then we discussed about the what are enantiomers and the diastereomers, and then we discussed about the epimers. If the two sugars have same configuration except on a one carbon atom, then they are said to be epimers. That means they differ from one another only at a particular carbon, but whereas the remaining the configuration on the other stereocenters is identical. So if the two sugars differ from one another in configuration only at a one carbon atom, then they are said to be the epimers. We can see here that the glucose and galactose differ at the fourth carbon, so they are C4 epimers, and whereas the glucose and mannose differ from one another only at the carbon 2, they are the C2 epimers, and so on. And then we discussed about, I think that's where we have stopped. Then coming to the cyclization of sugars. So most of the sugars, mainly carbon 5 and higher carbon 5 and more carbon containing sugars mainly exist in aqueous solution in their cyclic forms. And in the cyclic formation, the sugars either can form the pyranose structure or the furanose structure. In order to understand the pyranose and the furanose structure, we need to understand what exactly happens when alcohol reacts with the aldehyde. When alcohol reacts with the aldehyde, hemiacetals are formed. So during glucose cyclization, you can see here the alcohol group on the fifth position and on fifth carbon attacks the carbonyl carbon. As a result, the reaction is between the alcohol and the aldehyde, and the hemiacetal is formed. And here, in the same way, when alcohols react with the ketones, hemiketals are formed. Again, in case of fructose, the the same thing, the interaction between the hydroxyl group and the keto functional group gives the hemiketals. And in cyclization of sugars, what we need to understand that in aqueous solution, all the 5 are more carbon containing sugars mainly present predominantly in the cyclic forms. In the cyclization, either the sugar can form either 6-membered pyranose ring structure or the 5-membered furanose ring structure. And when during cyclization, you can see here that during cyclization, you can see here that the hydroxyl group on the 5th carbon attacks the carbonyl carbon. As a result, the cyclic structures are formed. And initially, in open chain form, that is Fischer projection, the C1 is achiral, whereas upon cyclization, C1 becomes chiral. And on C1, if the hydroxyl group is present on the down, then it is known as the alpha sugar. If the hydroxyl group is present on the up, then it is a beta sugar. So same thing for the glucose or the fructose and so on. Then coming to the anomers. Anomers are the 2 sugars which differ from one another in their configuration around the anomeric carbon are said to be anomers. For example, you can see here, this is as I already said, that glucose, when it cyclizes, it can form alpha D glucose or the beta D glucose and beta D glucose, and the alpha D glucose and beta D glucose you can see here. So in cyclization, if the hydroxyl group is present on the down, then it is alpha, it is up, it is beta. And if these 2 sugars differ in their configuration only at the carbon 1, this is also known as the anomeric carbon. Anomeric carbon are the hemiacetal carbon, and this linkage is known as the hemiacetal linkage. So that means anomers are the 2 sugars. They differ from one another in the configuration around the anomeric center is said to be anomers. So alpha D glucose, beta D glucose are anomers because they differ from one another only at the anomeric carbon. And now coming to the how to draw the Fischer projection. From the Fischer projection, I just wanted to give one example here. And before going to that, though these sugars mainly present in the cyclic form in the aqueous solutions, but because of the 6-membered pyranose ring cannot be planar because of the tetrahedral geometry of its saturated carbon atoms. Therefore, the sugars exist either in the pyranose ring exist either in the boat or the chair conformation. And in boat and chair conformation, all the substituents what we are seeing here, all these substituents can take up 2 orientations, either axial position or the equatorial position. Axial bonds are the perpendicular to the average plane of the ring, whereas the equatorial bonds are the almost perpendicular, almost parallel to the plane of the ring. And so that means because of the 6-membered pyranose ring cannot be a planar because of the tetrahedral geometry, and all the pyranose ring adapt into either chair or the boat conformation. So all the substituents can take up 2 orientations, either axial or the equatorial. So that means the sugars mainly exist in the aqueous solution. Mainly they exist in the chair conformation because chair conformation is more stable than the boat conformation. So that means the Fischer projection and Haworth projection is only to understand the stereochemistry and also the cyclization, but actually the sugars exist only present in their chair conformation. And now if you want to draw from Fischer to Haworth to the chair conformation, you need to understand that in a Fischer projection, when we are drawing the Fischer projection to Haworth projection at the cyclic structure, or the cyclic structure in the sense the chair conformation. So in Fischer projection, for example, I'm just drawing the glucose structure. This is a CH2OH. Here the CHO 1, 2, 3, 4, 5 OH OH, H, OH, and here H, OH, H. This is D glucose. This is the Fischer projection. So when it is cyclization, as I already said, that cyclization is between the hydroxyl group on the C5 and the carbonyl carbon. So when it cyclizes, we represent the cyclic structure in the Haworth form. So this is the cyclic structure of the glucose, and this is the carbon 1, 2, 3, 4, 5, CH2OH, 6. So now, how do we write the Fischer to Haworth? The simplest way to remember here is that the substituents that are present on the right side should be, because C1 and C5, look at here, this is C1 and this is C5, both are already involved in the linkage. So therefore we have only C2, C3 and C4. So that means the substituents that are present on the right side in Fischer projection should be written down in the Haworth projection. For example, here on the second carbon, OH is right side. That means OH should be written down. On the third carbon, H is down. H is right side. H should be written down. On the fourth carbon, OH is right and OH should be written down. Here OH is down means H is up. H is down means OH is up. OH is down means H is up, and here it is H. So this is here if the OH is down, then it is alpha, and in the same way, if the OH is up and the H is down, then it is beta. So that means it is alpha D glucopyranose, or we can also write the beta D glucopyranose. That means the conclusion here is that substituents here. So the substituents in Fischer projection, if the substituents that are the OH group is present on the right side, OH or H, anything, right side should be written down in the Haworth projection. If the OH group or the other substituent present on the left should be written up in the Haworth projection. So, and here coming to the C1, on C1, if it is OH is down, it is alpha. On C1, if the OH is up, it is beta. So this you need to remember, that to transform Fischer projection to the Haworth projection. So if the Haworth projection is given, you should also be able to transform into the Fischer projection and this one. So as I already said, that sugars mainly present in the chair conformation, and so when you are writing the chair conformation, again you need to see that the substituents that are down in Haworth, let me write it as here chair conformation. Substituents that are down in the Haworth should also be written down in the chair conformation. Substituents that are up in chair conformation should also be written up. But we don't say in chair conformation, we don't say up and down. We generally represent either axial or the equatorial, as I already said, that axial or the equatorial. So now let us you can just see here that I will just draw the structure of the glucose with the chair conformation. When you are writing, you should first draw the and as I already said that we have, this is the carbon 1, this is the 2, this is the 3, and this is 4, and this is 5, and of course this is CH2OH, this is 6. So now, as I already said, that all the substituents can take up two orientations: axial, equatorial, and of course it is axial. So all axial bonds are sorry, axial, 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 equatorial, 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 equatorial. So all these axial and equatorial bonds are on the opposite side on adjacent carbon atoms. Now you can see here on the C1, for example, we are just writing the chair conformation of the alpha D glucose. Here the OH group is down, so here it should be written in the axial position. Axial position, that's down. Then OH down, here OH. Then H down, here H. Then OH down, here OH. Then of course here H. So when OH is down, obviously here H. When OH is down, here again H. If the here H means again here OH, here OH means again here H. So this is the chair conformation of alpha D glucose. And one more point I need to highlight that, as I already said, that in all D sugars, the bottom stereocenter, sorry, this is the bottom stereocenter, C5 carbon, the bottom stereocenter, the hydroxyl group should be present on the right side. Then only they are said to be D sugars. If the OH group is present on the left side, then they are said to be L sugar. So in case of L sugars, the CH2OH group, that is here on this points down. So that is the thumb rule to know whether a sugar is D sugar or L sugar. In the Haworth projection, if the CH2OH group is into the ring, you can see here. CH2OH group is into the ring, then it is known as L sugar. And as I already said, that D sugars are only naturally abundant, not to the L sugars. So this is about the how to transform Fisher projection to the Haworth projection, Haworth structure, Haworth to the chair conformation. So now coming to the muta, I think many of you have already know what is a mutarotation. Mutarotation, it is nothing but interconversion of one monosaccharide, one anomer into the another anomer. For example, when D-glucose, alpha D-glucose, when it is dissolved in water, and initially the ring opening takes place and it goes into the open chain form, and ring closes again in a different orientation, and it also forms the beta D-glucose. You can see here here it is alpha D-glucose, and this is a beta D-glucose, and these three forms are in these three forms are present in the solution. So that means conversion of one anomer to the another anomer through open chain form is known as the mutarotation. And of course, it also this can be when the one anomer is converted to another anomer, the optical rotation also changes, because freshly prepared glucose, alpha D glucose, has plus 112 degrees, whereas the beta D glucose has only plus 19 degrees. So that means in aqueous solution, the one anomer is converted into another anomer through open chain form, and during this the ring has to open and the ring has to close in a different orientation, such that the H and OH on the anomeric carbon goes on different ways. In the sense, in alpha D glucose it is in the down, whereas in beta D glucose it is up. So in order to push the OH up and H down, the ring has to open and the ring has to close in a different orientation. So this interconversion of one anomer to another anomer is known as the meta rotation. And coming to the sugar derivatives. So in the living organisms or in cell, in addition to the monosaccharides, there are many sugar derivatives are also present. They have various physiological functions, mainly the amino sugars like galactosamine, mannose-amine, and other sugars like glucosamine, and certain sugar derivatives like glucose 6-phosphate, and then glucuronic acid, and gluconate, and gluconolactone, and acidic sugars like sialic acid. All these are the sugar derivatives. They have many functions in the cell. And this is about the how to the cyclization of sugars, and also how to transform the Fischer projection to the chair Haworth form, and Haworth form to the chair conformation. Then coming to the, and all of you please remember, if I give any chair conformation, and if I ask you to draw the Haworth as well as the Fischer, and if I ask you to identify what this sugar is, you should be able to do that. And for that, what you need to remember here is that you need to remember the shortcut what I have given you, and the get Raxel AAGM GAGT in the same way. The another shortcut that is said DRX PFST. These are things you should remember. Then only you will be able to understand. You need not memorize any structure. Once you remember this one, once you remember the diglyceraldehyde structure and the dihydroxyestone, then you will be able to do the any structure of the aldose family or the ketose family. So all of you, you need to practice the all these structures. It is very, very important in the examination point of view. Then coming to the reactions of monosaccharides. So, and it is not that important, but there are many reactions we can study from the monosaccharide. But I will be explaining only the 5 reactions, and which are important for in the biochemistry point of view. First one is the formation of glycosides. Formation of glycosides, in the sense of we said that when the glucose in the glucose, this linkage is also known as hemiacetal linkage. Hemiacetal linkage. This is also known as the hemiacetal carbon. When any monosaccharide, when it reacts with alcohols, and glucosides are formed. You can also see here, this is when beta D glucose upon reacting with the methanol, it forms the methyl beta D glucopyranoside. So here, this is the methyl group is added here. So this is known as the glucosides. And you please understand that beta D glucose, when we dissolve it in water, the ring opening can take place, and ring closure can take place in a different orientation. As a result, beta D glucose would be converted into alpha D glucose through open chain form. But the minute when it is if it is in the glycoside form, that is here it is methyl glucoside, and the ring opening is not possible because it doesn't have the free anomeric carbon, whereas the sugars have the free anomeric carbon, whereas the glycoside doesn't have the free anomeric carbon. Therefore, glycosides doesn't exhibit the meta-rotation at all, because in order to exhibit the meta-rotation, one anomer has to be converted into another anomer through open chain form. In order to do that, the ring opening and the ring closure should take place. But whereas in case of glycosides, the ring opening is not possible because it doesn't have the free anomeric carbon. So therefore the glycosides are generally glycosides cannot exhibit the meta-rotation. And this is about the glycoside. You can also see here. You can see later. Second one is the reduction. When sugars undergoes some reduction in the presence of sodium borohydride or the other reducing catalyst, and the sugar alcohols are formed. You can see here, this is the glucose, and when it is undergoes reduction, the carbonyl group, aldehyde group, is converted to the alcohol. This we know that from alcohols upon oxidation gives aldehydes. Aldehydes upon oxidation gives carboxylic acids. In the same way, carboxylic acids upon reduction gives aldehydes. Aldehydes upon reduction gives alcohol. Same thing happens here. So here it is aldehyde, and when it is reducing, again it is becoming alcohol. So that means glucose in the presence of sodium borohydride gives glycetol. This is polyhydroxy alcohol. The aldehyde group is reduced to alcohol. And in the same way, different sugars can form different alcohols. And for example, glucose forms glycetol, galactose forms galactitol, and it is also known as the dulcitol, and mannose gives the mannitol, and of course fructose gives both sorbitol and mannitol. So that is about the reduction. Reduction gives the polyhydroxy alcohol. Then coming to the oxidation. So sugars undergoes oxidation and gives various products, and in the presence of mild oxidizing agent like bromine water, like bromine water. In the presence of mild oxidizing agent, you can see here that in the presence of mild oxidizing agent, the aldehyde group undergoes oxidation to carboxylic group. Therefore, glucose gives gluconic acid. And in the agents, even primary alcohol group also undergoes oxidation, and the CH2OH becomes a COOH, and they are known as the aldaric acids. So that means glucose gives, in the presence of bromine water, it gives gluconic acid. In the presence of strong oxidizing agents like nitric acid, it gives the glucaric acid. They are also known as the aldaric acids. And in addition to this, in the human body, and because of certain enzymes, the glucose also undergoes oxidation by in uronic acid pathway, and gives the glucuronic acid. In this glucuronic acid, the CH2OH group is oxidizes to COOH. So you remember here is that, as I already said, that when aldehyde group is available, chemical reagents like any oxidizing agent cannot convert CH2OH group to COOH without affecting the aldehyde functional group, because as I already said that hydroxyl alcohols upon oxidation gives aldehydes upon oxidation gives carboxylic acids. So now when you oxidize, when the CHO, when it is oxidized, it gives the alcohol. So but when it is oxidized, it gives the carboxylic acid. So here, in the presence of mild oxidizing agents, only aldehyde group is undergoes oxidation to carboxylic acid. In the presence of strong oxidizing agent, both aldehyde group as well as the primary alcohol group undergoes oxidation to the carboxylic acids. But in the human body, in uronic acid pathway, and the glucose undergoes oxidation to glucuronic acid, where the primary alcohol group undergoes oxidation to carboxylic acid without affecting the CHO. Because this transformation is not possible by any chemical reagent. This can only happen only by the in the cell, in the uronic acid pathway. And of course, these glucuronides or any other uronides, they have various functions in the cell, mainly in the removal of the toxic substances in the xenobiote, in the removal of the toxic substances. So this is to the oxidation. So oxidation in the oxidation, sugars in the presence of mild oxidizing agent, strong oxidizing agent, and in uronic acid pathway, they form a gluconic acid, glucaric acid, glucuronic acid, respectively. So then coming to the last reaction, that is the dehydration reaction. So when monosaccharides, when treated with the concentrated sulfuric acid, the 3 water molecules would be removed and forms the cyclic aldehydes, and these cyclic aldehydes are known as the peripherols. So glucose gives hydroxymethyl peripherol, whereas the 5 carbon sugars gives only the peripherols. And so this is about the peripheral, and what we do in anthrone reaction in the lab, biochemistry lab, based on the formation of the peripherols at the beginning. This peripheral further only condense with the anthrone reagent and forms a peripheral anthrone in order to determine the total sugar present in a sample. And the last reaction is the Wassermann reaction. It is also known as the Wassermann formation. And the monosaccharides, when they react with the excess of phenyl hydrogen, they form Wassermanns. These Wassermanns are the crystalline compounds, and they are used for the identification of sugars, because these Wassermanns are the crystalline compounds. Different sugars have the different sugars can form different Wassermanns, and they have the different shape of the Wassermann crystals. And you can see here that the important point is that in Wassermann formation, only carbon 1 and carbon 2 are only involved in the reaction. That means if the sugars having similar configuration on C3 to C6 can form the same Wassermann, because during Wassermann formation, the stereo the configuration of the C1 and C2 would be lost. Therefore, sugars having the similar configuration, same configuration on C3 C4, C5, and C6 can give the same type of wasazone. And this is the overview. And sugars, monosaccharides undergoes oxidation to either the aldonic acids or aldaric acids or uranic acid. Upon reduction, gives the polyhydroxy alcohols, sorbitol, mannitol, and so on. In the presence of dehydration, gives the pyrrols, and in the presence of excess phenyl hydrogen, gives the wasazones, and the wasazones are used for the detection of the sugars qualitatively. And of course, these are the reactions what I had already explained. The same thing you can go through. These are the sugar alcohols. Different alcohols upon reduction gives different sugars, upon reduction gives different alcohols. Glucose gives glycerol, mannose gives mannitol, xylose gives xylitol, and so on. And these are about the of monosaccharides. These are important. This one. Now coming to the disaccharides. Coming to the disaccharides. So in oligosaccharides, again we have oligosaccharides are further classified into the disaccharides, trisaccharides, tetrasaccharides, and so on, depending upon the number of carbons, the number of monosaccharide units present. And in these disaccharides, two monosaccharide units are held together by glycosidic bond. You can also see here, this is one maltose, and the two glucose units are joined by the glycosidic bond. And what you are seeing here is that this is the glycosidic bond, and these glycosidic, you can also see here, this is one glucose and this is the fructose, and when the sucrose is formed, water molecule is eliminated and the glycosidic bond is formed. So in the same way, and you can also see here the glucose and glucose, when they condense, water molecule is eliminated, as a result the maltose is formed, and the linkage, this linkage is known as the glycosidic linkage. And based on the carbon atoms which are involved in the glycosidic bond formation, all these disaccharides are classified into 1,2 glycosides, 1,6 glycosides, and so on. And we will the important glycosides, what we study here is that the disaccharides, one is maltose. So maltose is composed of two glucose residues, and these two glucose residues are connected by the alpha-1,4 glycosidic bond. You can see here, this is the alpha-1,4 glycosidic bond. This is the carbon 1 of the first sugar. This is the carbon 4 of the fourth sugar. Since here it is the, when the linkage, here it is the alpha glycosidic linkage, alpha-1,4 glycosidic linkage. So this is, since the second sugar has the free hydroxyl group or the free anomeric carbon, the maltose is a reducing sugar, and it can also exhibit the mutation. So this is about the maltose, and it is composed of glucose, two glucose units with connected by the alpha-1,4 glycosidic bond. Coming to lactose, lactose is composed of the glucose and the galactose, and of course the galactose is the first residue here, and these two are again the galactose and the glucose residues are connected by the beta-1,4 glycosidic bond. You can see here, this is the first carbon and the fourth carbon. Here this is the linkage is the beta-1,4 glycosidic linkage. Beta-1,4 glycosidic linkage, you can also clearly see here, galactose and glucose both are connected by beta-1,4 glycosidic bond. And since lactose also has free anomeric carbon on the second sugar, it is also reducing sugar, because it has a free anomeric carbon, and so it can be in equilibrium with the other anomer through open chain form in the solution. And coming to the sucrose, if you see the sucrose, and you can carefully see that sucrose is composed of glucose and fructose, and the linkage is alpha-1 beta-2 glycosidic bond, alpha-1 and alpha double-headed arrow beta-2 glycosidic bond. In the sense, the anomeric carbon of the first sugar and the anomeric carbon of the second sugar, this is in this case this is the ring. In order to connect these two, the ring is inverted. This is the first carbon of the fructose. This is second. This is the third. This is the fourth, and this is the fifth, and this is the sixth. So that means the linkage is alpha 1, beta 2. That means the anomeric carbon of the glucose and the anomeric carbon of the fructose both are anomeric carbon of the fructose both are involved in the glycosidic bond formation. So therefore both the anomeric carbons are involved in the glycosidic bond formation. So therefore it doesn't have the free anomeric carbon, and the sucrose is not a reducing sugar, because both the anomeric carbons are involved in the glycosidic bond formation. And in addition to this, we have other disaccharides like isomaltose. In maltose, the linkage is alpha 1,4, whereas in isomaltose, you can see here, this is isomaltose, the linkage is alpha 1,6. In the sense, the first carbon of the glucose and the sixth carbon of the second glucose both are involved in the glycosidic bond formation. So that's why the isomaltose has the alpha 1,6 glycosidic bond. And cellobiose also again is composed of glucose and glucose and beta 1,4 linkage. It is again similar to in maltose alpha 1,4 cellobiose. Two glucose residues are connected by the beta 1,4 linkage here. And trehalose, and again it is very, very important here. You can see here, of course, this is for the sake of representing the glycosidic bond, the ring is inverted and also rotated. And here, this is the first carbon of the glucose and the first carbon of the glucose, and this is also the first carbon of the second glucose. In the sense, trehalose is a disaccharide composed of two glucose units connected by one double-headed 1,1 linkage. In the sense, anomeric carbon of the first sugar, anomeric carbon of the second sugar, both are involved in the glycosidic bond formation. So that's why, since both the anomeric carbons are involved in the glycosidic bond formation, the trehalose is also not a reducing sugar. And again, of course, the trehalose is a major sugar present in the insect hemolymph, and also it is also a reserve food metal in the certain fungi. And these are about the various disaccharides. And in addition to this, we have I think raffinose, that is a trisaccharide, and that is composed of the glucose, galactose, and the sucrose with alpha 1,6 linkage. This you can see later. So this is about the disaccharides. Polysaccharides, we will discuss this one in tomorrow's class.